The large amount of genetic diversity that exists on continental Africa is unique, but also it means the variations between each region within Africa is also quite large. So we cannot simply say that one region is representative of the whole of Africa, because we know now that is not true. In our study, we have found genetic risk markers or factors for prostate cancer across 3 regions that is unique to Africa. And we were only able to achieve that by studying African populations here on our continent. And going forward, I think it is important to continue to study African populations for diseases that is relevant in Africa. Genomic technology is very expensive, especially in our setting when there's competing health priorities. It is very difficult to say where you're going to spend your money to get the best outcome. It's impossible for one scientist or one team, one group to do everything. So we need to be able to collaborate as wide as possible and to work with experts and field leaders on these topics, because then it saves us a lot of times in the learning curve itself. We don't have to reinvent everything. We can leverage on all the skills that was been built before, and we can apply it in our specific setting.
It may sound simple, but a good way I always like to start conversations like this is to look at what led to this. What were you thinking? What was the team curious about? Even before starting the study, what were you hoping to find going into this study?
Absolutely. Thank you for this opportunity. We know that prostate cancer disproportionately affects men of African descent. Now, when I say that, I particularly refer to men of sub-Saharan origins. These are continental African men, that in our particular setting, prostate cancer is the most diagnosed cancer in men not in worldwide, but in Africa. Disproportionately, our mortality rate for African prostate cancer in Africa is actually much higher than anywhere else in the world. Yet the amount of study that's been conducted in Africa is somewhat limited. Previously, genetic studies into prostate cancer, the genetic understanding the genetic risk factors that leads to increased prostate cancer incidence has been mostly conducted in men of European ancestry, and disproportionately very little research is done in Africa itself. This is what led to the foundation of the consortium, is really to establish a network of prostate cancer researchers across Africa. One, to increase the capacity to do research within the continent, and 2 is to better understand what is the driver behind this particular phenotype that we see.
Okay, thank you very much. Curious question: why do you think prostate cancer is understudied on the continent, despite the fact that, to some extent, we can't say it's not popular in terms of people being aware of it, but why the study gap?
So, there are several very complex factors that leads to why it's understudied in Africa. Historically, scientific research in Africa itself is somewhat limited. In the context of genetic studies, there is a very nice website that actually curates the number of large-scale genetic studies that looks at genetic risk factors for certain diseases. Now, we're not limiting this to cancer, and there you can see number of studies conducted in Africa or on resident African population counts for a very small fraction of what has been done across the world. And in our context, I think funding is a big challenge. These kind of studies, large-scale consortiums, are very expensive to fund, very expensive to run. And in Africa, I think there's a lot of competing priorities in our setting that naturally would prioritize infectious diseases like HIV, tuberculosis in the past. And cancer is not, or I wouldn't say it is not as highly prioritized as some of the other more pressing issues.
Okay, thank you very much. Before I talk about the study, I'm curious about the consortium. How was it formed? What went into bringing everybody together?
Yes, so that I can speak to the historical knowledge of what I know. I joined the institution after the consortium was formed, and I really need to give credit to my predecessors at both the National Cancer Registry of South Africa as well as the University of Witwatersrand, as we are the main one of the main participating centers in this consortium. So our consortium is called the Men of African Descent Carcinoma of the Prostate Consortium, or abbreviated to MADCAP, MADCAP. And this consortium was formed back in 2016. It has really started with a group of researchers and clinicians on the continent that wanted to better understand what is this driver behind the increased incidence of prostate cancer, and how can we better understand this condition, and ultimately how can we better manage it. The consortium was funded through a National Institute of Health initiative, the NIH funding mechanism from the USA. The first round of funding was funded through what we call a Year One mechanism. That might be a little bit too complicated, in the sense really just a form of funding that allows for multi-country collaboration. And how the consortium was set up is that from the multi-centers across the continent, we have, if our recollection serves, we have 7 academic hospitals and research institutes from 5 African countries. That is each paired with a counterpart in the US, a institution that is based in the US, one to study prostate cancer in all aspects, from the clinical aspect, from actively enrolling new prostate cancer patients, and then to do genetic studies, to do clinical studies, to do epidemiological studies. The partnership there really is aimed at strengthening partnerships and as well as strengthening skills, capacity development and skills transfer from the US into Africa to allow this kind of research.
Yeah, thanks for that clarification. Just a quick follow-up. What is peculiarly unique about studying prostate cancer that we can't expand current system for studying other cancers in Africa too?
So in the context of our study is the largest of its kind in Africa. When we say that what we mean is that this is the largest genome-wide association studies of prostate cancer in continental residents of African men. This has not been done at this scale in the past. Previous genomic studies into prostate cancer, particularly relating to any participant of African origins, largely African American based study cohorts, and the rest is largely European centric cohorts that has been established. With our cohort, it is the largest of its kind at this point, which is under 4,000 prostate cancer cases and 3,500 non-cancer controls of ethnically matched controls, to allow us to look at the genetic risk factors behind prostate cancer. Now, similar study methodologies are currently being undertaken across the other common cancers that we see in Africa, particularly in South Africa. Our research group also works on breast cancer and esophageal squamous cell cancer, as well as cervical cancer. Those are also cancers of high burden and high importance, which we're conducting a similar study design on, hopefully to uncover the genetic risk factors that leads to the increased incidence of these cancers.
Yeah, thank you very much. I think let's talk about the selection of the countries, of the study sites in the 3 African regions that were included in the study. Was there a rationale behind these regions? And let's talk about the countries too.
So we know that African genetic diversity on the African continent, just in the population itself, is extremely diverse, with African population being some of the oldest on the world. The large amount of genetic diversity that exists on continental Africa is unique, but also it means the variations between each region within Africa is also quite large. So we cannot simply say that one region is representative of the whole of Africa, because we know now that is not true. So historical studies that looked at African Americans as a proxy for continental African populations, we have learned that now with new evidence and new data in literature say that is not true, and each region within Africa needs to be studied by itself. So this is why it drove us to the 3 representative regions across Africa, which is East, West, and Southern Africa. In terms of the country participating, I think there it was a combination of previous partners which we have worked with in the past through the existing research collaborative network, as well as countries with really high burdens of this cancer that will allow us to be able to amass a large enough cohort to be able to study it. One of the challenges of our particular study approach is that we need a very large study population. So that gives us enough statistical power to identify the risk factors that we're looking for.
Okay, thank you very much. What about samples, number of samples collected from these different countries? Are they equal in terms of the representation, or are we getting more samples from a particular country more than the rest? I'm suspecting probably South Africa has more representations because of the location, but I might be wrong. So that is what I'm trying to figure out.
So in terms of West Africa versus Southern Africa, we have relatively similar ratios, if I recall. But if we look at individual sites, the Wit site definitely contributed the most amount of samples that was enrolled over the study period. This is largely due to the burden and the case volumes we see in our clinics, where at the Baragwanath Academic Hospital in the Department of Urology, the outpatient department, we see a lot of prostate cancer patients compared to anywhere else on the continent at this stage. I think that is perhaps speaking to the burden we see in South Africa itself, but as well as the unique nature that our tertiary healthcare system, or rather our public healthcare system, is free for all population to access within the country. So that's why perhaps we see a lot more patients seeking care. But that is beyond my expertise.
Thank you very much. So let's talk about what was done. So you did whole genome sequencing for?
No.
Okay.
No, so we did genotyping using a novel it's not a novel microarray in the sense that one of our consortium goals that we developed a microarray that is specific to capture African genomic diversity. We also know from existing literature that some of the off-the-shelf available options for microarray genotyping may not capture African genetic diversity well enough. So as part of the goals of the consortium, that we developed a microarray chip that specifically enriches for African genomic diversity, so we're able to capture this diversity using this array, and also enriched for the known prostate cancer risk regions that's been identified in previous studies.
Interesting. So let me push my luck a bit, whether I understand what I'm trying to ask right now. Inputting, if the goal is to identify the uniqueness of the unique loci for the African continent, that means the development of the microarray chip would also be very, very unique. How were we able to test in building up the target on the microarray chip that they will be unique for the African continent, or were they already identified targets on the unique in previous, or let me say, where did the data for the development of those targets that the microarray chip was picking up come from, if we are now using it, or is this a two-stage study that you did?
So, to my knowledge, the development of the microarray was based on existing available African genomic data that has been published through other initiatives that may not necessarily be related to any cancer work. But the general principle of genome-wide association studies that we leverage on something called linkage disequilibrium. What that means is that group of genetic markers may be inherited as a chunk of the chromosome. So if I try to put this in very simple terms, we have 23 pairs of chromosome that makes up our genetic constitution. Of the 23 pairs, we inherit half of the chromosomes, with 23 chromosomes, from our mom, and the other half from our dad. And as those chromosome pairs meet and they form the cell that grows us into a fully formed human, and our reproductive cells form thereafter, there's a process called genetic recombination that happens. So whereby the the paired chromosomes meet up and they swap pieces of each other. They swap the pieces of chromosome with each other in the aim to create genetic diversity. And that is, from an evolutionary point of view, strengthens a particular species going forward. Now, I'm putting this in very, very simple terms. The older a particular population is, the more chances of genetic recombination of this particular kind of recombination have happened. Thus, would lead to more genetic diversity. If you look at a population like the European population, which they migrated out of Africa, their degree of diversity is much less when you compare it to an African population. Now, for a genome-wide association study, what we're leveraging on is that we're leveraging on genotyping one or two markers in a particular region that will be able to help us identify all the other relevant markers in that particular region, on the hypothesis that that chunk of chromosome is passed on together onto the next generation. And in Africa is particularly advantageous is that we have much larger degrees of diversity in the African setting, that we can actually what we call fine map, zoom in into the regional genome with a lot more granularity to identify the risk markers that is potentially contributing to the phenotype. And the reason why off-the-shelf solution does not work is that when you design a genotyping microarray chip, you always most of the design starts with a genetic backbone. Now, these backbones are developed from existing sequencing data to capture large regions within each chromosome so that you can pull down sufficient genetic information. But we know that in an African setting with a higher diversity, we need to capture more regions to be able to capture this particular diversity. I think that is the principle behind why we went to build a new chip, in order to capture these diversity. At the time when the chip was developed, I believe we leveraged all the amount of African data that was available publicly at that point in time. And then subsequently, we compared the performance of that array that our consortium has designed against any off-the-shelf solutions. And we noted that it definitely performs better in African population by comparison.
Thank you for that extensive explanation. And so let's talk about the results. As somebody that worked on the team, so I would like you to talk about it before I zero in on interesting aspects of that I quite find interesting. So for you, I know a lot of data were generated, but at your fingertip, what really stood out in your results?
So the key findings in our results at this stage, we identified 3 loci or genomic regions with a strong association with increased prostate cancer incidence. These 3 regions are located on chromosome 8q24.21, 6q22.1, and 11q13.3. All 3 regions were previously quantified in other studies for prostate cancer, but what's unique in our particular setting is that the lead signals within each of these regions we have found in our study were unique to African populations. Meaning, if we didn't study our African population, we would never have been able to find these markers. And this also talks to the advantages of studying African genomes, that we have much higher levels of diversity that allows us to map or zoom in finer into a very large chromosomal region to identify the potential causal variant that's driving up the increased risk. And the second key finding we found is that although all 3 regions showed very strong association with increased prostate cancer incidence, the amount of contribution of the risk between each region, so east, west, and south, varies a lot because of the genetic diversity that we see on the continent.
Sorry, I was muted. I was already talking. So let's talk about the relevance of this broad diversity. But before I talk about it, I think in the result you talked about that the genetic architecture was differ across Africa. Does this mean that the architecture in Nigeria was different from in Ghana, was different from Senegal, was different from South Africa and Uganda? Or are there some countries that we can say, okay, the architecture, even though they differ across the continent, these 2 countries are similar? Or do you have different architecture for each country studied?
Can I share by any chance?
Yes. Let me see if you can share. You can share, I think, screen.
Because I think if there's a way to share, I think that would be great.
If you see share,
Let me see tab
yes.
window. There we go. I found it. Great.
Okay.
So I think what answers your question best is really looking at this figure B over here. So in simplicity, what this figure is, if you look at going across the rows are the three continental regions from west, east, and south. The darker the color, the stronger the association signals from each site. And at the bottom, each column is a genetic marker that is of relevance that we found and marks the three regions, 6q22, 8q24, and the 11q13. So as you can see, east and west shares a very strong signal in the AQ24 region. The signal is present in southern Africa, but to a lesser degree. And if you look at the other two signals, 6q22 and 11q13, you see something very similar. So that talks to really the diversity we have across the continent. Populations within the western, or rather in a geographical context, so in the western part of Africa, are largely more similar with each other than they are to population from east or southern Africa. That talks to the historical migration patterns of population within Africa. So we do definitely see a very large degree of diversity across the larger regions as opposed to smaller geographical regions. From South Africa, we had two participating centers. One is University of Stellenbosch, that's enrolled from the Western Cape, and then us at Wits, we enrolled from Johannesburg. The populations within Southern Africa or South Africa were genetically similar to populations that are in East or West Africa.
That's quite interesting. So, what are these genetic differences? If we talk about the manifestations of these differences, what does that translate to prostate cancer disease? Or how do you know what these particular targets encodes for, or what the effect could be? Does it make it a higher risk disease? Does it make it a higher virulent disease, or what are the impact on the patient?
So by study design, our work looks at the common genetic variants that contributes to a particular phenotype. So if we look at, by and large, germline genetic contributions, or the inherited components that contributes to any phenotype that we see, can be loosely categorized into two groups. There is the rare variant part. The rare variant part, we're looking at very, very rare mutations in functional genes that we see in an individual or in a particular population that has a direct impact to a particular phenotype. The best example I can think of right now is the BRCA genes in breast cancer, as well as coincidentally in prostate cancer, where a mutation in one of the BRCA genes, a germline mutation, directly affects the protein function of the protein of that particular gene, thus leads to a substantially increased risk of developing cancer in that particular context. So those are largely referred to the rare variants. In our particular study, we looked at the common variants, which these are variations that occurs within a population at a relatively higher frequency. So here we looked at, I think, if I recall, it's about 1% frequency or more. Each of these markers, from a statistical point of view, contributes a very small amount of risk to the phenotype. Each of these markers by themselves will not be able to drive any manifestation of a particular phenotype, but the cumulative effect of all of these markers will be able to push an individual into a higher or a lower risk bracket. So studies from elsewhere in the world have demonstrated this tool to be very powerful in the sense to develop something called the polygenic risk score. So we genotype a large set of genetic markers, like what we've done in our particular study, and then we identify the markers of interest that is indicating a difference between cases and controls, and we quantify the cumulative effect of all of these risks for a particular individual, then we're able to confidently stratify individuals either into a high risk group, intermediate risk, or a low risk group for a particular cancer. And other studies in the past have demonstrated that individuals who's classified into the high risk group would actually benefit from advanced or additional interventions, such as more regular screenings or consults or follow-ups that will be able to assess if they are acquiring cancer at any point in their life. So that talks directly to the clinical management aspects of this particular phenotype. And in our particular setting, in our study at this point in time, this is really the very first step in a lot more work to come going forward, is that we're hoping to be able to develop or adapt a context-sensitive risk stratification method for prostate cancer in our setting, then use that as a triaging tool to be able to better manage prostate cancer risk in our patients.
Yeah, thank you very much. Then, how well distributed are these types in the population? Are they very, very unique, or are they only found in few individuals that could make of high or low epidemiological relevance?
So in our study, in the manuscript itself, we did try to quantify the AQ24 haplotypes, and then if I can share again, there we go. So you're looking at particularly at figure B over here. We're looking at the AQ24, and then really the size of the circles indicates the commonality or the frequency of that particular haplotype occurring in our study population. And the darker colors, I'm not sure if it's very clear on your side, the darker colors indicate the cases and the lighter color indicates the controls. As you can see, some of the more common haplotypes, the distribution really it's roughly half-half between cases and controls. But there are some really interesting haplotypes that we see that are less common in the general population, but also has a very, very distinct difference in the distribution between cases and controls, particularly if you look at haplotype 3 over here of this particular branch on top right, and then also this 11 and 12, you can see these particular haplotypes occur a lot more frequent in cases compared to controls. Then potentially this haplotype here is of interest, that it is tagging the chunk of the chromosome that is driving up the risk in our setting.
Well, that's quite interesting. So, how do you see this data being applied now? So, what can we currently use these data sets that you've generated for?
So, all of the studies data set that we have generated so far from the study has been made publicly available, of course, through the relevant data access policies. So this is currently available on dbGaP, if I'm not mistaken. The summary statistics from our study is available on the GWAS catalog, that you can easily find the findings itself. And in terms of its implementation, we have several other ongoing studies that's stemming from this work that's really looking at the finer regions to hopefully identify what is driving this particular increased association that we're seeing in our study. Additionally, aside from doing the microarray data on our entire cohort, we have subsequently went on and did whole exome sequencing of a subset of our study populations. There, the research question is asking what are the rare variants that we see on continental Africa that's potentially driving up the phenotype that we are seeing, and that work is currently ongoing at this stage. In terms of the clinical and epidemiological aspect of it, the consortium is still going. We're in our second round of funding at this stage for the next 5 years. And we are prospectively enrolling prostate cancer patients with a slightly shifted focus in looking at aggressive prostate cancer in the African context, and how do we apply new molecular diagnostic techniques in our setting to be able to identify aggressive prostate cancer from the other prostate cancer types. As you know, about a third to half of prostate cancers are slow growing prostate cancer that is unlikely to cause any adverse health effect during the patient's lifetime. But 1/3 of the prostate cancers that we do see, and we're also seeing this now with some clinical evidence that happens more commonly in Africa, is that 1/3 of it is very aggressive prostate cancer, where a patient could actually benefit from more aggressive treatment strategies as well as earlier times of intervention. So the second 5-year funding for our consortium now is just transitioning onto that aspect. And then, so as soon as we have more findings, we'll be very happy to share.
Thank you very much. And I think probably I saw that the goal initially from that abstract section was to infer ancestry-specific genetic architectures. Were there other genetic architectures that were discovered that did not fit into this particular category of the architectures that you were looking for that may also be of importance?
Sorry, can you just repeat that question? I couldn't catch all of it.
Okay. So I said, from what I can read from the paper, is the aim was to infer ancestry-specific genetic architectures. Were there unique, non-ancestry-specific genetic architectures that were also seen in this study that are of relevance?
At this stage, not. At this stage, we are seeing African-specific genetic architecture that exists with unique differences across all three regions. That is important in our context. Going forward, our main message is that we need to study high burden diseases in Africa using African populations. We cannot simply infer findings from elsewhere to the continental Africa. That translation is not beneficial. Instead, we should be aiming to drive and build research capacity in Africa itself, and then conduct the studies here to find what's driving these conditions in our setting.
I think you are where I would like to round up, which is sharing insights from this experience about the reality of doing something like this on the continent for other high burden diseases. So for other high burden diseases, which we know there are several of them, that the knowledge gap still is very, very. What is currently available now that can make closing this gap to be realistic at this moment?
So through our network itself, we aim to do a lot of capacity development where possible. And one of the other highlights of our study is that majority of the analysis was done in Africa. All of the genotyping was done in a facility in Cape Town. We made sure to keep all the wet lab laboratory on the continent. Bulk of the analysis was shared between us and our collaborating center in the US. And that way it facilitated a lot of skills transfer. And, not only looking at prostate cancer itself, but there are other large African consortiums at this stage that is driving the very same thing that we are, is to enhance African research capacity, to bring genomic research to Africa, to empower African scientists to take this work forward. One of the good examples I can think of right now off the top of my head is the Africa Consortium that's been running for several years now, and they are one of the largest African consortiums that has been established over the past few years, with that very specific aim of looking at common non-communicable diseases in Africa, and to see what are the factors that's driving it.
Yes, thank you. I think there's also this very, very relevant and connected issue, which is getting the best and the most out of data generated. For me, I think there is a wide gap between researchers like you and entities that could get the best and the most out of the data, especially translating them into probably tools or like developing testing kits, or probably unique treatment options. And so what is currently the model or the approach to handling this data? I know you say it's publicly available, but do you think putting them at the public alone is a right strategy, or do you agree with me that there should be better collaboration. For instance, let's say the genetic startup landscape on the continent that could look at the data generated and look at insights or tools that can be generated from that instead of reinventing the wheels, starting intending to actually generate their own data. I don't know whether you understood my question.
Absolutely. And I do agree with you. Data sharing is the first and foremost point here. So all data that's been generated, I mean, a lot of the funders now these days, to my knowledge, requires all of the genomic data that they have funded to generate to be made publicly accessible. That way, it's also to conserve resources, just that the same researchers don't repeat the same work again without accessing any existing data. Collaboration is key. At this point in time, our group is working to populate the foundational knowledge aspect of it and hopefully lay a strong enough foundation that that allows us to take our work forward to future goals, as you mentioned, to be able to build a tool that we can implement in our local setting. Genomic technologies is very expensive, especially in our setting when there's competing health priorities. It is very difficult to say where you're going to spend your money to get the best outcome. But at the same time, these kind of foundational research is important in our setting, because only by conducting these kind of studies can we build up that knowledge base to take our work forward in future to potentially access a technology at a later point that is beneficial at a much wider scale. Then again, it comes back to collaboration. It's impossible for one scientist or one team, one group to do everything. So we need to be able to collaborate as wide as possible and to work with experts and field leaders on these topics, because then it saves us a lot of times in the learning curve itself. We don't have to reinvent everything. We can leverage on all the skills that was been built before, and we can apply it in our specific setting. And ultimately, I think a collaborative research environment is beneficial for everyone involved. And one part of it is very important in scientific communication is how do we get this very complex knowledge that we have found now to be translated to a level that is easily accessible to the general public, and so that the key messages can be understood and that knowledge can be spread.
Yeah, I think that's fabulous. So, if you have, I think this is my last question before asking for your last remarks. If you have power to pull off anything that you like, what do you want? What impact or what difference or what do you desire to happen to these results that you've generated?
That is a grand, ambitious goal. I mean, in South Africa, we work in a reality where there's the private and public healthcare sectors that serves our general population in the country. And the amount of disparities that you see across the two sectors is very striking. And when we look at our collaborative partners across Africa, healthcare is not a universally free access elsewhere on the continent. But I guess if you were to ask me, what is the one thing that I could achieve, and to achieve it really is to be able to provide equitable healthcare across the entire population, across the entire continent. I mean, there should be no disparities between seeking healthcare, or rather accessing healthcare. But in reality, it is very far from that. So I guess that is where I would lean towards.
Interesting choice. So I always like to do this close up with this, which is in as few words as possible, simplify this research to let's say to a 10-year-old kid whose parents are not geneticists and may not have been hearing these things gradually. Is it possible for you to attempt to simplify this study for a 10-year-old?
Okay, let's try. I would say we know that genetically, population in Africa is extremely diverse. And now in our study, we have found genetic risk markers or factors for prostate cancer across 3 regions that is unique to Africa. And we were only able to achieve that by studying African populations here on our continent. And going forward, I think it is important to continue to study African populations for diseases that is relevant in Africa. And more importantly, we need to build more skills and capacity in Africa to allow African scientists to carry out this kind of work going forward.
That was a nice time. That was a nice try. Thank you very much. So any last remarks for me?
No, that's all. Thank you so much for this opportunity to publicize our work. We're extremely excited about this, and we're also extremely excited about where this could potentially take us going forward. So thank you so much for this opportunity.